Each HIV virus consists of 2 copies of its RNA genome and a surrounding layer of proteins called the capsid. Each individual protein is called a capsomer. The virus's genome is located inside the capsid, which is surrounded by a lipid bilayer viral envelope with virus-specific protein protrusions called spikes. The glycoprotein responsible for attachment to a cell is GP120. HIV's viral specificity results from GP120, which recognizes the specific CD4 receptor sites found on macrophages, brain cells, and primarily T lymphocytes. The attachment alters the shape of the bound GP120 and CD4 proteins, allowing a different portion of GP120 to attach to the co-receptor CCR5 or CXCR4. Attachment causes GP120 proteins to spread out and reveals GP41. GP41 extends from HIV and docks with the cell's membrane. It then pulls the HIV pathogen and cell together, fusing their lipid bilayer membranes and releasing the capsid into the cytoplasm.
Cellular enzymes dissolve the protein capsid, releasing the 2 copies of the genetic RNA and the 3 replication enzymes. The enzyme reverse transcriptase converts the information into DNA by adding complementary DNA nucleotides to the viral RNA, forming a DNA-RNA duplex. Ribonuclease H breaks down the RNA portion of the strand. The cell's natural replication process then creates the corresponding complementary strand of DNA. HIV integrase binds to the newly formed HIV DNA in the cytoplasm and cleaves each 3' end. The HIV DNA complex moves into the cell nucleus through a nuclear pore complex. The HIV 3' hydroxyl end binds to the host cell's DNA 5' end. Fully integrated HIV DNA is called proviral DNA. Using the cell's normal process of protein creation, mRNA corresponding to the proviral DNA moves back to the ribosomes in the cytoplasm for translation to a specific amino acid chain. The enzyme protease cleaves the amino acid chain into portions that correspond to structural proteins and replication enzymes. The structural proteins then surround complete viral RNA strands and the 3 replication enzymes, forming a capsid that buds out of the cell, taking with it a portion of the cell's membrane and spike proteins.